Okay, at long last, here's the final part of C1.1. Please excuse, the way that I sound. I'm sorry, I'm a little bit sick. And so, oh, whoa, please focus. I think that's not focus. Please fix yourself. Okay, so enzymes' effect on activation energy, and this is really just, what is activation energy and how your enzymes will help to increase it, or not increase it, but decrease the amount of activation energy needed to increase reaction rate. And so, woof. Effect of enzymes on activation energy. Whoa. Most chemical or biological reactions require energy in order to occur. This energy is called activation energy, so it's just that required energy for a reaction to occur. With the help of enzymes, reactions can be carried out with a lower investment into activation energy. So with the help of enzymes, reactions can be carried out with a lower investment into activation energy, or less activation energy, and this will allow more product to be produced, and the rate of the reaction is increased. And so you have this little model here that's showing the breakdown of lactose into glucose and galactose. Without an enzyme, you need a lot of activation energy in order for this to happen. Just to get it up to the point where that reaction occurs, you need a huge amount of energy. With your enzyme helping, so by binding that substrate to its active site and starting to weaken those bonds, the amount of energy needed to get it to the point where that reaction can occur is much less. And so it makes it more efficient for your body, and it also makes it a lot faster. So, activation energy without enzyme is this whole huge thing needed, whereas activation energy with enzyme, just this little bit. I should have picked two different colors. Oh well, it's too late now. And so, without your enzyme, you have much more needed. With your enzyme, boop, boop, you have much less. And so what is activation energy? It is just the what have I done? Minimum energy required to start a reaction and it's lowered by enzymes, or by enzyme activity. And so your next step says energy is required to reach a transition state before completing a reaction. What in a chemical reaction is the transition state? And so it's like that intermediate before being converted into your products. So it's like when the bonds are starting to be weakened, but they haven't quite fully formed the product. They're in kind of like a weird, reaction limbo. And so your transition state is your intermediate. No, let me spell intermediate right. Intermediate before being converted into products. So it's like your enzyme substrate complex. This would be your transition state. Or just like when the bonds are weaker because energy's added, but it hasn't quite gone yet. This would be like your transition state. Transition state. And so some examples. When bonds are broken or weakened in a substrate. And this bonds being broken and weakening of your substrate is like what the activation energy is used for. I guess I'll write that in orange. That way it matches all the other activation energy stuff. This is what activation energy is used for. Boop. Converting your substrate to its transition state. And again, your enzymes, they do a lot of that for it so that it can reduce this activation energy needed for the reaction to occur. And so an enzyme lowers the activation energy by weakening bonds already. Duh, that's what we just talked about. And therefore, less energy is needed for an exothermic reaction to occur. After the biotics have been formed, energy is released. And so, let me get the whole picture. And so what you're looking at really just pretty much how enzymes affect this. So enzymes aren't going to affect the energy that's released from it. Actually, oh shoot, I have to scooch this back up. Sorry. So if we look both with and without your enzyme, you have the same amount of energy released as a result. Energy released. The only thing that's changing is that activation energy, that energy needed to start. What's released with your enzyme is not going to be changed. And so, when you're using an enzyme, I don't know, what color? We'll use pink, I guess. The total energy in a reaction, let me straighten the page, okay. So, total energy released isn't changed. Only activation energy is reduced. And so, to kind of list out like the steps of this little process that you're seeing here, I don't know. We'll do a fun gradient from green to blue to purple, because I think I have 3 things. So your enzyme catalyzes the reaction. Oh, how do I spell catalyzes? Catalyzes reaction when your substrate bonds or binds to active site and is altered to reach. Oh, we have a color for transition state. Yay! Transition state. And so this is when your enzyme binds. You have your initial amount of activation energy added. Oh, I guess we should also have like activation energy added. And so then after that, hello? That's right, I was gonna do blue. After that, your reaction occurs. So like the little arrow for then. And your products are made. And released from active site. And any like excess energy is also going to be released. So it's like change in free energy. I guess we'll do and energy. So you have your activation energy added. You have your enzyme come in here. Woof. It gets us to that transition state. There we go. And then your reaction occurs and your products and other energy are released. And then again, without your enzyme, it's way higher for the activation energy. Alrighty, on to our next step. Let me flip my little guide sheet. These are the notes that you should have taken from your enzymes and industry presentation. Please, somebody remind me, send me an email if I don't have those posted on Canvas yet by like Tuesday or Wednesday. Like, please tell me, because I know I'm gonna forget it. I think the only thing that like needs to really be added to this page, besides the notes you took in class, some key things about enzymes and industry that I think either I neglected to like have you all cover, or just like didn't really fit in your presentations. That's also like important for you to know. Enzymes in industry. Are typically immobilized. Let me spell immobilized right. Immobilized. Or like bound to some kind of membrane. And this is that they can be easily separated from the reaction so they can stop it at the ideal time to produce the maximum amount of product without any sort of negative effects. And so they can be easily separated, stopping reaction at ideal time. And then I think the other like thing that if you didn't already put these pieces together yet is that enzymes have diverse functions. Which allows them to have diverse applications. That feels like really obvious, but you know, you never know. It might only be obvious to me because I'm old. Who's to say? And then again, these ones are from your presentations in class. Please, somebody remind me if I don't post those slides to Canvas, because they were actually really nice. Here are some practice questions for you. The practice questions, because I think that these are fairly easy to see. I can show you like what color each one is supposed to be in just a second. But then they also tell you how to calculate your measurements and enzyme reactions. Sorry. This is something that I think is a little bit tricky to like do without doing, like to explain to you without like having you do it. And we're gonna practice. I forgot to have us practice this in class. It's been a very scatterbrained week for me. But so when measuring the enzyme catalyzed reactions, you measure the amount of substrate that has disappeared from a reaction mixture, or the amount of product that has accumulated in a unit of time. And so that's basically what you're plotting. So you're going to plot the amount of product over time, or the amount of substrate that's disappeared over time, either way. And so your shape of your curve is what's going to indicate your reaction rate. However, it's not constant, like it's going to change. Like as you notice, it starts out faster and then ends up kind of leveling off, being slower. And so that like change is not constant, like the gradient, the rate is not constant. And so if you were to calculate the rate of an enzyme reaction, you'd have to calculate it for a particular time. And so in order to do that, you would go to the time that you wanted, and you would find your change in product over your time at that point. So it's pretty much just like the slope of a tangent line. It's how you calculate it. And then initially when you calculate it, it's 0. I feel like I did not explain that very well. I'm sorry. But so Your rate is not constant, and so it can only be given for a particular value of time. And so to measure it, you draw a tangent to the curve, or like a line on the curve at the specified time, and divide the change in product by the change in time. Or like essentially just the slope. Like the rise over run, the slope of your tangent. So here's your tangent at your specific time. And then you do your product over your time. So the formula would be, reaction rate, or I guess, what do they call it? Instantaneous rate of reaction. Oh, jeez Louise. Instantaneous rate of reaction equals the change in product over the change in time. That's really it. It's not difficult, but I feel like it doesn't really make much sense explaining it without like an example problem. And this example problem is like not the same. And so give me 1 second and I'll pull up the one in color so I can match up the lines for you so that you can do your little practice questions. I have it open somewhere. La la la. How's everybody's Monday? Hopefully pretty good. Click and hello. Nope, it's in this one. Gosh darn it. Okay. Hang on. Bear with me. Stay with me. I don't really think you have a choice. It's video. Okay, there we go. Okay, maybe these were harder to look at than I thought. Sorry. So I'll tell you all which line is which. So for 51 degrees Celsius, it is this, hello? It's this bottom one? That doesn't make a ton of sense. This data is not making a lot of sense, but that's okay. For 4 degrees Celsius, it is this one, apparently. For 21 degrees Celsius, it is this one. Oh, never mind. I guess that makes sense. I take it back. I lied. Sorry. And then, what other color is going to show up different? We'll do pink. And then for 34 degrees Celsius, it's this one. I don't really care if you all can do conversions, so I would focus on trying A, so calculating the mass of starch in grams, calculate the mass of starch per minute, convert per minute to per second, I don't really care. Explain the variation in oxygen percentage at time 0, use the graph to determine the rate. And then I think that's fine. The other ones, I'm not too worried about if you can do them or not. But if you can try on your own to do A, C, A, and B, that would be fantastic. Oh, and these are 2 different questions. This one's about this one. This one's about this one. All right. On to the next set. Oh, shoot. That's the wrong way. La, 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 la. Is this the penguin one? Yay! It is the penguin. Sorry. I was waiting on this one. I'm very excited about it. Can you tell? Oh, I need to get my other set of pens. Listen, this is why I was excited. I get to use my glitter pens. It's a rare occasion. Anyway, so this is talking about generation of heat through metabolism. And this is some of the stuff that is like HL specific. And so heat, of course, has to be red, duh. So generation of heat, energy by reactions of the metabolism. And so metabolic reactions often release heat. As a byproduct almost, and this is because the products of the reaction generally have less energy than the reactants, sorry. And so that additional energy is converted to heat. So that heat that's released at the end of a reaction is usually just like waste energy. And so it's converted to heat just so that it can like be removed from the body. And there's different mammals and different, I guess not just mammals, but like different birds, mammals, fish, whatever, that have all found ways to use these to help conserve heat. And so the one key thing to note, we'll write it in red because it's heat, is that your conversion of energy is not 100%, which is why you have this loss of heat. Woo, causing loss of heat. And then here's your fun little man. And so, how is heat generated? By the reactions in metabolism used by birds and mammals. And so, because the products have less energy than the reactants, excess energy is just released as heat, which can then be used to help maintain internal temperatures. So, products have less energy than reactants, excess energy is released as heat. Used to maintain internal temperatures. Internal temp. And so the little examples they give yours are cute. I like this little penguin. And so in very cold conditions, emperor penguins will huddle together in groups to take advantage of the metabolic heat released by neighbors. So it's like they get to share their little heat together. And so you have these little fatty penguins here, and see how they're all kind of clustered together, except for these ones who I'm sure are freezing their little patoodles off. And then this picture I feel like is not done justice in black and white, which is why, we can color it in together. Get excited. If you have like a highlighter, that'll probably work. If you don't have a chaotic, amount of like glitter metallic pens. And so your lower heat, it's like 20-ish degrees and less, we'll make purple. And so you can see at their like extremity points, they have your coldest temperatures because there's not a lot of metabolic activity happening. And you can also see like on the little backs that are exposed to the cold and aren't like tucked inside of a hot like penguin huddle crevice. I'm so sorry. That you have like colder bits. I looked at the color image of this to figure this out, by the way. This is not just like Miss White can read temperature through a black and white image. And then as we get a little bit warmer, from like 20-ish to like 15-ish, still not super warm, but you can see that you have your coolish temperatures on the outside. Again, remember a lot of your metabolic processes are digestion based and like in your thinking area, like your brain. I'm sorry, I'm like losing my mind. And so you'll see a lot of your colder spaces around the edges. You can also see colder bits on your penguins, again, where they're not like tucked away in a little penguin crevice. It's like on their little exposed backs. This guy's like really cold, and look at him go. This guy's freezing, oh my gosh. And then as we continue to heat up, we get to like, oh, that's the wrong side. Sorry. Stop it. We get to like our 10-ish to 15-ish range. We start getting towards the middle, where some of those metabolic processes are happening, where your penguins are kind of tuckled together, so they have their little hot penguin crevices, they're sharing that heat. Look at him go, sharing, oh, I colored over that guy's nose. Oh well, it's fine, he'll be okay. And so when they're tucked in together, they're sharing that heat, they're staying warmer. when we get, this is like a small range for some reason. I don't know why. I swear to you I copied this from the color image, so I don't know why the ranges are like this, but this is like half of the way to 5. But then we start to see a lot more where they're nice and warm because they have either a high amount of blubber there, they have their high metabolic processes, especially in our penguins that are like cozied up. They have these like little crevices of hot penguin area where they're sharing their heat, and so they can be nice and warm, nice and hot. Little hot penguins. And then their highest temperatures, no, it's the wrong side again. Gosh darn it, Jessica. Again, are going to be like right in the middle of their tummies, like where they have the most blubber and they have the most heat on their faces because their little brains are cooking away. And then in these little nooks and crannies, like between the penguins where they're all nice and wedged together and they're sharing that warmth. These just look like blobs now. Oh well, it's fine. So again, they'll have that hotter temperature, either where they're doing more of that metabolism, where they have more metabolic processes going on, where they have higher blubber, and of course in their hot penguin crevices. It's like when you like snuggle together with your bestie for warmth when you're camping in the winter. Why would you camp in the winter? I don't know, I'm making it weird, I'm sorry. A lot of different animals also have this brown fat or that adipose tissue, which is what we talked about when we looked at loons and seals. And so in addition to behavior, things like huddling, like the little penguins to get their little hot penguin crevices, little penguin pocket. Oh my god, a penguin pocket. Okay, sorry. Birds and mammals also have other adaptations to their metabolic rate to help raise their external temperature. And so this is a picture of a white fat cell and a brown fat cell. Your brown fat has more mitochondria, so that it's doing more cellular respiration and it's having more energy generated and then lost through heat to help warm up the body. You have your brown fat, there's some in like your shoulders, and then there's some around like your main organs to help keep you warm. And again, I'll do a little heat map of these bad boys for you. So like there-ish. And you have your coldest bits around the outer edge of your white fat cells because they're not specially adapted like brown fat. It's slightly warmer, very slightly, on the inside of your white fat cells because that's where a lot of their metabolic processes are happening. It's not exposed to that like external cold. It's like almost like wearing a jacket, how your inside's warmer. Or like how your fingers get coldest when you go outside in the cold. Maybe that's just me, I don't know. And like your mid-range temperatures, you have like a really small bit around here because it's being hit by the external cold temperature. And then as you go inward, as you have those mitochondria in the center, as you're having that heat stored that's being released, you get warmer and ultimately warmest. Hello? There we go. Warmest. And so where's the pen? And so brown fats in adipose tissue contains more mitochondria than white fat. Oh, white fat and any other tissue for that matter. Meaning that more energy is generated through lost heat during uncoupled cellular respiration. An uncoupled cellular respiration is like cellular respiration that they're kind of just doing for heat. Like no ATP is actually produced. It's just happening to do this heat generation. It's a special thing your brown fat does. You also have involuntary muscle contractions. I feel like this page is going to be all red, so we'll switch it to like pink. Think shivering. It generates more heat and therefore raises body temperature. You're doing cellular respiration to get that energy to shiver. When you're shivering, when your muscles are contracting, you're releasing heat as a result of those processes not being fully efficient. And then here's a fun one. So decomposing manure or compost can become really hot, as seen by this steaming heap. They literally put a steaming pile of shit in the packet. I think that's really funny. And so what organisms are responsible for this metabolic heat generation? It is your bacteria and other decomposers, but mainly your bacteria. Breaking down the organic matter is a metabolic process. Kind of like digestion for bacteria, which releases heat as a byproduct. Oh, and I get to have so much fun. I finally have a use for my stupid, ugly brown highlighter. I can color in my poo-poo. Wow, I feel so special. My brown highlighter, you finally have a purpose. Look at you go. Do you feel so much less useless now? I don't know what this white glitter one does, but we'll put white glitter on the steam. Let's see what this even does. Oh, almost nothing. Okay, well, that was fun. On to our next page. Oops. No, I think that is the right way. Yes, it is. So now we're going to talk about enzyme inhibition. We'll make enzyme inhibition, we'll make it purple. And so this is what we looked at with our little Ed Puzzy. We didn't get to touch on this as thoroughly as I wanted to, but it's okay. I'm hoping that this will hopefully help catch us up a little bit. And so some chemical substances reduce or even prevent enzyme reactions from happening by blocking the enzymes. That would be like your competitive inhibition. And the substances that do this are inhibitors or... Why does it say inhibitors or enzyme inhibitors? That's really dumb. Okay, anyway. And then enzyme inhibition can be competitive or non-competitive. And so it can either be an enzyme that, or like a substrate that binds to the allosteric site, which just denatures the enzymes that your substrate can't bind. Or in competitive inhibition, it's a substrate that binds to the active site so that your substrate itself can't bind to that active site. And so these chemical substances are called inhibitors or enzyme inhibitors. Why? Okay, whatever. It's fine. And so we'll make a competitive blue. And we'll make non-competitive, I don't know. I'm tired. I was going to make it blue and red to make purple, but I'm tired of looking at red, so we'll do green. The green one still has blood on it. Okay. So we have competitive inhibition, where it's binding to that active site. There's the inhibitor. And we have non-competitive inhibition, where it's binding, to an allosteric site, or like an additional binding site on the enzyme. What color should the enzymes be? I threw all the stupid colors everywhere. Okay, we'll make the enzyme yellow, and the substrate, you know what, we'll let the brown shine through again. Stop it, I can have another win. And so our substrate will be brown. So normally, it would bind on to the active site. And this one, our brown substrate, tries to bind, sucks to suck. Competitive inhibitor's already there, can't do it. And then our substrate here tries to bind, but oh, non-competitive inhibitors here. Shape's changed, can't do it. Sorry, buddy. Gotta go. Can you tell? Okay, I'm having too much fun. All right, so then our enzymes will make them yellow. Boop, 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 boop. There it goes. Look at them go. Oh, I should have just done the active site. Oh well. It's too late now. We'll have to just label the active site. I'm not going to use the yellow pen. That's a mistake. We'll make it orange. So here's, ooh. So the active site. Active site. Oh, this one was already labeled. Okay. Why were none of the other ones labeled? Okay, here's the active site here. Active site. That says active site. I promise you. And then active site here. This one's denatured active site. Denatured active site. Alrighty. So, how fun and exciting. Let me not mess up my own color key. Okay. And so non-competitive inhibition, make it blue. Whee! Dun, da, da, da. I have no self-control. We'll also make its little example down here. Spoiler! Make its little example blue. I have a love-hate relationship with how they put the similarities in the little line in the middle. Like, it makes sense, I get it, but it kind of interrupts my color plan. And then we have competitive inhibition. Oh, it's time to draw another square. Wee. And then we'll do our little example. Spoiler again. It's an ACE inhibitor. Wee. And then similarities. I guess they'll be purple. The colors are they're not vibing today. It's okay. I did it to me. Okay. Which one to talk about first? What pen do I already have out? I already have green out, so we'll talk about competitive inhibition first. So, in competitive inhibition, your inhibitor and substrate are really chemically similar. Inhibitor and substrate are very chemically similar. Remember that they have to go to the same active site, and a lot of how your active site is paired with its substrate is through those chemical similarities. We need to refresh, flip back a couple pages. And so your inhibitors that are going to go into your active site and compete with that substrate, they are going to need to be very chemically similar so that they can bind to that same active site. Speaking of which, inhibitor binds with same active site as your substrate. And it causes your enzyme to not be able to or cannot bind the substrate. Because your active site is occupied. It's like if you were to wait in line for the bathroom, but then all the stalls are occupied, then you just gotta wait. Similar. The substrates are waiting in line for the toilet, but the active site's already been used. I like to think of it like To remember the difference between competitive and non-competitive, the inhibitor competes with the substrate for the active site. It's like they're racing each other. They're going to beat each other up for it. For active site. You spell active site right. There we go. And so if we were to look at our little picture, oh, I need the brown one again. Oh well, it's okay, I'll get it in a second. So we have our enzyme. We'll color him yellow. Color our enzyme yellow here. Whee! Color our enzyme yellow here. Whee! And our enzyme yellow here. And so we need, where's the brown? There we go. What are these substrate shapes? Okay, whatever. So we have our substrates. Okay, there we go. All right. And then our competitive, oh, this one, no, it's okay. And then our competitive inhibitor, look at it go. I kind of hate that it's just a square. Boop, boop, boop. And so, where is the pen? And so step 1, you see here, your competitive inhibitor and your substrate, they both compete for this active site on your enzyme. There's your little active site. Oh. Step 2, your inhibitor binds at that active site, and therefore your substrate can't bind. It's very sad. Step 3, you have your substrate here. No, hello? Already bound in. It is reversible. And so once you have your substrate at a certain concentration, your inhibitor will leave, and your substrates can bind again, and they're nice and happy. Oh, speaking of which, let's give these ones little faces. Hello? Where's the black? This substrate's hyped. He's like, oh, what is under here, pen? He's like, heck yes, I can bind. This one's like, oh no, see you later. Stinky poo-poo, can't bind. And this one's like, oh no, see you later. Stinky poo-poo, can't bind. He's got to go. Okay. Like losing my gosh darn mind doing this. Okay, where's the blue? I don't want that blue. I want the other one. So for non-competitive inhibitors, your substrate and your inhibitor aren't they can be chemically similar, but they usually aren't. So substrate and inhibitor, my throat hurts, not chemically similar. They bind at different active sites, so they don't really need to be chemically similar. It's better if they aren't, because then this inhibitor will go straight to its little allosteric active site. Oh yeah, this is also known as allosteric inhibition. You'll hear these terms used kind of interchangeably, because like the enzyme with the allosteric site is called an allosteric enzyme. The other site that's not your active site is called the allosteric site. And so if you hear allosteric inhibition, it's the same as non-competitive. Why did they write competition here? They should have just put inhibition, but whatever, it's fine. All right, your next thing, or how it works, is your inhibitor binds to a different site, your allosteric site, and changes shape of active site. One key thing is that the substrate might still bind, but much slower. or not at all. Depending on how changed it is, sometimes the substrate can bind and the reaction rate will happen, but it's much slower. Or your substrate will sometimes bind, because remember, it has to have that perfect lineup. And so having this even a little bit distorted is going to make it harder for that perfect collision to happen in the first place. And so if not stopping it altogether, it'll at least make it like significantly slower. And so our allosteric site, we'll mark it on all three. Before I put this pen down, I will color our enzymes. Whee! Whee! You have your allosteric inhibitor, or your non-competitive inhibitor, which will bind to that other site. As a result, when it binds, that active site changes, and then where's the brown? Your little doo-doo substrate can't bind anymore. He's very upset. He'll get a frown too. Well, where's the black? There it is. Oh, he's gonna be angry. He's pissed. He's like, what the heck? I came all the way here to bind to this gosh darn active site. Here's the active site. Active site, normal, binds. Active site, goofy. All right. I'm so sorry. All right, let's look at our similarities. So both of these are reversible. So both of these, your inhibitor, when it's no longer needed to inhibit that enzyme activity, can be removed, and then your enzyme function will return with both your allosteric inhibitor and your competitive inhibitor. So reversible, reverse. How do I spell reversible? Able. And they both also slow or stop enzyme activity. Sorry. And so your two examples down here for competitive inhibition, you have ACE inhibitors or statins. And so normally, where's the green? Sorry. Normally, angiotensin is a substrate produced by an enzyme, your ACE enzyme, which binds the receptors in the wall of your arteries, inducing constriction of blood vessels and an increase of blood pressure. ACE inhibitors will inhibit the action of this enzyme to control high blood pressure by preventing the binding of your ACE. And so this is really common in medications. It's common that you'll get like an ACE inhibitor as like a pill or like a statin as a pill to try to lower blood pressure. It prevents high blood pressure. Oh, how do I spell pressure? Pressure. Hypertension / heart failure. How do failure? Right? Failure? Okay. And then with your scooch, stop it, get out of the way. With your allosteric inhibition, these are all like poisons, which is kind of, I don't know, I just find that really interesting. And so things like heavy metals, so mercury, lead, or copper, inhibit the activity of a wide range of enzymes, such as dehydrogenases. Heavy metals inhibit the activity of a wide range of enzymes, like lactate and malate dehydrogenase, and digestive enzymes like amylase, by irreversibly, key thing, binding to your allosteric site and preventing binding of the substrate. This is the case in all kinds of heavy metal poisoning, resulting in a wide range of symptoms. And so the reason that they're irreversibly bonding is because they form strong covalent bonds. Covalent bonds. And this is like specific, where am I going to put this? Okay, that'll just go weirdly up the side of the page. It's specific to enzymes with sulfur containing R groups Those sulfur-containing R groups are really causing problems. They're the only way disulfide bonds occur. They also cause, like, the ability for heavy metals to bind inseparably and cause a bunch of problems. Speaking of the problems, you have a wide range of impacts when you have heavy metal poisoning because so many of your enzymes have this sulfur-containing R group somewhere in their allosteric site or on a side of the enzyme. And so it can impact your mouth, your abdomen, your reproductive tract, reproduction, your brain, pretty much anything neuromuscular. So it's crazy. Crazy. And it's permanent. So, kind of a nightmare. Alright, on to our next page, which I think is looking at graphs of... Oh, nope, I lied. The graphs are later. Sorry. This is just looking at the examples we just looked at, kind of. Well, one of them is. I don't know. Listen, I do these pages in order when I make my little guide, and so I write the notes as I go. Alright, so statins as competitive statins as an example for competitive inhibition. It's competitive inhibition, so it'll be green. And so cardiovascular disease is a major concern in the world, as it ranks one of the highest causes of death. In most cases, it's a buildup of cholesterol in the blood vessels that's responsible for increasing blood pressure and in long run causing damages to the heart and cardiovascular system. And so drugs known as statins, which are prescribed by doctors to patients with high blood pressure, act as a competitive inhibitor. And so it's essentially what we just talked about, but I'll break it down for you step by step-sies. Let me get the pen. Hello. And so do some research to find out how statins work to help treating the problem of cardiovascular disease. Joke's on you. I already did it. Sorry. I'm talking to the packet, not like you. And so it's competitive inhibition. It treats cardiovascular disease caused by high cholesterol, cholesterol. And so what your statin does is it binds to an enzyme. So statins, did I spell statins right? Yes. It looks too much like Stalin, and so it's like goofing with me. Sorry. Statins bind to an enzyme, specifically HMG CoA reductase, if you were curious, that naturally synthesizes or makes cholesterol. No, we're supposed to synthesize. Is there a Z? Sure. Synthesizes cholesterol in the liver. So it prevents additional cholesterol production in humans who, in humans who already have high cholesterol. Something that's like really important is correct dosage. Important. to lower to the correct amount. As one thing that we don't want is to like lower it too much and then not have any cholesterol, or have like super low blood pressure. Like your cholesterol, although in high amounts is a problem, in small amounts it's natural and it's like good for you. And so you just don't want to have a ton of cholesterol reduction. But in people who already have like a high buildup of cholesterol, like from diet, I guess like parentheses from diet / like, I don't know, malfunction in regulation, lowering that naturally produced cholesterol can help to even out the levels. That way you're not like overloaded with it and you're not seeing those negative effects. Alrighty. Bump it up. And so now we'll look at mechanism-based inhibition. This is not non-competitive inhibition, but I don't want to make it green too. We'll make it purple. It's penicillin. P for penicillin, P for purple. I don't know. I'm sorry. So mechanism-based inhibition. And so many antibiotics act by binding irreversibly to the active site and inducing a permanent change. And so penicillin binds to the bacterial enzyme transpeptidase. Read the article on the right to answer the questions. This article is printed incredibly small, so read it if you can. And so it says, so far we have learned about forms of inhibition which are reversible. What makes this type of inhibition irreversible? And so this is another example where you have that formation of your strong covalent bonds. So that formation of strong covalent bonds with a sulfur containing amino acid in the active site. So it binds to the active site and it like locks in. It forms those covalent bonds. It's not going anywhere. Because it binds to the active site, what type of inhibition is it? Drum roll. It's competitive. Woo! And so competitive. We know that because it binds to the active site. Oh, the green snuck its way in here. And so, what is the consequence of binding penicillin to transpeptidase? Consequence of binding penicillin to transpeptidase. It's kind of like a chaos tale of what happens to these cells. And so, the enzyme transpeptidase, when bound with penicillin, how do I spell penicillin? It's written on here somewhere. Oh, did I already mess it up? No, I didn't. Penicillin is unable to build cell wall. And so transpeptidase is building your cell wall. When penicillin binds, it stops it from being able to build cell walls. But the enzyme that breaks the links and starts to break links in the cell wall is just part of how it's built, is still going. And so without the cell wall being able to be replaced, the one that's cutting through it keeps going, and it creates these holes, and eventually your cell wall will shed. And then because the pressure within the cell can't be held together by just the membrane, your bacteria bursts and dies. There you go. Enzyme that breaks cross links, parentheses cell wall, keeps working, which leads to creating holes in the cell wall and eventual shedding of cell wall, causing cell to die by bursting under pressure. I do not think this is the image they really should have picked. Alrighty, now on to the graphs. So, interpreting graph displays of enzyme activity and the effect of inhibitors. I'm gonna make that title purple. Stop it. Not you, this. So, interpreting graphs and displaying enzyme activity effect of inhibitors. And so, we'll look at graph 1 and then graph 2. Graph 1. These are some spoilers. But so graph 1 is showing you competitive inhibition. And so with different percentages of the inhibitor, these also need to be colored. Okay, that's fine. I'll fix it for you, I promise. Oh, okay, there we go. So with different concentrations of the inhibitor, you have different rates of activity. And so I'll show you what that looks like in just a second with the colors. And then your other one, graph 2, is non-competitive inhibition, because with different concentrations of the inhibitor, your enzyme activity follows a really similar path. Regardless of how much inhibitor, it just is like the overall effect. And so, what's my best plan of action here? High amount, low amount. I guess yellow will come out to play for once. And so 0% inhibitor is going to be this top line and this top line with the fastest rate. Boop. 25% inhibitor. No, the lights are turning off. Gonna be this middle line. And then 50% inhibitor. This is going to be this bottom line. And so, what does graph 1 show? Competitive inhibition. What does graph 2 show? Click. Non-competitive inhibition. This video is getting really long. I'll try to pick it up. I'm sorry. I've been dilly-dallying. Actually, you know what? In my defense, I don't think I've been doing that much dilly-dallying. This one's for graph 1. And so what we're looking at in graph 1 is when we have a high substrate concentrations, so lower inhibitor concentrations, your substrate outcompetes your inhibitor for your active site. So again, imagine they're like trying to race each other to this active site. If there's more of your substrate than there is of your inhibitor, you're not really going to see a ton of effect because those inhibitors aren't binding as frequently as your substrate can. And so we see a very low impact on the rate, although it's still some, because some of those inhibitors are going to beat them there. At higher substrate concentrations, we see a much less, we see much less of a change than we do with higher inhibitor concentrations. And so regardless, your rate of your reaction is going to increase and reach the same maximum as it would without the inhibitor, because again, they're competing for the same spot. If you have more substrate than you do inhibitor, your inhibitor is going to outcompete it. And so the amount of inhibition from a competitive inhibitor is going to be determined by the amount of substrate. And so let's put that ramble into words. So high substrate concentrations, which would be at like 0% and 25%. Your substrate outcompetes. How do I spell competes? Outcompetes the inhibitor for the active site. And so your rate of reaction increases and reaches the same maximum as without the inhibitor. Amount of inhibition depends on amount of substrate. And then we'll look at graph 2. Where's the blue? Burp. And so graph 2, no matter what, your non-competitive inhibitor is always going to significantly lower that rate of reaction, regardless of how much of your substrate there is, because it's not like binding for the active site. It's always going to be able to bind to that binding. It's not competing for that active site. It's always going to be able to bind to that allosteric site. And so your maximum enzyme activity, like this top corner of the graph, will not be reached if there's an inhibitor present. So non-competitive inhibitor always significantly reduces reaction rate. Your maximum enzyme activity will not be reached. That's because your allosteric binding causes deformation of active site, preventing binding, and can happen even if substrate is in the active site. Like, it doesn't have to worry about where the substrate is. It's always going to happen. I wouldn't worry. It's not like a real thing. Well, it's a real thing, but it's not like living. All right. And so the amount of inhibition is independent of amount of substrate. What? Amount of substrate. Oop, there you go. How many more pages of this do I have left? My throat hurts, and this video is taking forever. Okay, we only have 1 page left. I'm sorry, I promise I do like you guys, but it's late, and I'm tired, and my throat hurts. No, the light's turned off. It's okay, I'm not going to get up. I'm just going to turn the lamp on. We only have 1 page left. It's not worth it. All right. So our last page is about end product inhibition. I guess we'll make this one, I don't know. We'll make this one this color. So regulation of metabolic pathways by feedback inhibition, or end product inhibition. And so enzyme pathways can be controlled by the concentration of products from the end of a pathway. End product inhibition usually involves allosteric enzymes and non-competitive inhibition. And so we'll have your substrate, you have your little metabolic pathway, you have your end product, and then that end product will go back and it'll bind to an allosteric site, causing your enzyme to not create or bind any more of this substrate. And so, let me switch this to the other side. So your end products are here. Hello? And then here they are bound to the allosteric site. We'll make the enzyme, I think this is the yellow one. Make the enzyme yellow again. And we'll make the substrate, where's the brown? I don't know where the brown one went. Oh, there it is. And then we have our substrate. Beautiful. And so when your enzyme isn't active, your substrate can't do this metabolic process, and it's inactivated by that end product binding as a non-competitive inhibitor and stopping it from binding this. And it usually happens when you're trying to control the amount of end product, like you only want to make the amount you need to conserve and to save energy, which I think will make more sense when we look at the little example. So here's, end product inhibition in the isoleucine biosynthesis pathway. Woo! And so threonine and isoleucine are essential amino acids. What does that mean? It means they are, I guess we'll do essential amino acids. They are necessary amino acids that, there we go, hello, cannot be made in high enough amounts by the body. So in high enough amounts, they are needed from your diet. And so, hello? There we go. And so what is the function of threonine? What is the function of isoleucine? Look at this one. So threonine is involved with the formation of proteins in tooth and animal. Collagen and elastin. I spelled collagen wrong. And elastin. It also metabolizes fat and prevents buildup of fat in the liver. And it helps to enhance immune system. You know, I really thought these pens had a notable difference in color. I'm realizing now that they don't. It's fine. And so, what is the function of isoleucine? It's used in your Krebs cycle. During respiration. It can be converted into fatty acids. And special note, it can be made in the body from threonine. And where are they found? Cottage cheese. The only thing I've written in giant letters is cottage cheese. And then smaller, cottage cheese, poultry, or like chicken, fish, meat, lentils, beans, and sesame seeds. So eat cottage cheese, beans, and sesame seeds. Like, what the heck? Okay. So I'm looking at your little diagram. Where exactly does isoleucine bind to? And so this diagram shows you the entire process. So in high enough quantities, isoleucine binds to the allosteric binding site of the enzyme deaminase, preventing further binding of threonine to the active site. So because your threonine can be used in 5 steps to make isoleucine, okay, we need to assign these a color, otherwise it's going to drive me insane. So I was trying to avoid it, but no, it's not possible. They'll be yellow and green. So your threonine and your isoleucine. And so your threonine can be converted to your isoleucine. And so that process is requiring a lot of energy and requires a lot of steps. And so it's really only done if your body absolutely is out of isoleucine and needs some. And so you have your initial amount of threonine. You have your threonine in your active site. There we go. It goes through all these intermediates, and then your end product is your isoleucine. Your end product inhibition happens when your isoleucine binds to an allosteric site on that first enzyme to stop that from happening. So when there's enough isoleucine, it binds, and then it slows down this reaction rate and eventually will stop it. When your isoleucine is all used up by your cell and your cell needs more, it goes away from its little allosteric inhibitor, and then it starts this process to make more. And so, looking at the diagram, what enzyme does it bind to? Oh, we'll make enzyme 1 this color. Enzyme 1. There's enzyme 1. This is also enzyme 1. Here's enzyme 1 again. And so it binds to enzyme 1. Threonine deaminase on an allosteric site. And so, oh, well, you can't see that. Great job, Jessica. So, good enough. It's binding to an allosteric site. Here's your allosteric site here. And we'll make it the active site. There we go. There's your active site. Active site. Your active site. Remember, we have the induced fit, so it can scooch a little bit to let it in. And so, what type of inhibition is this? It's not binding to the active site, so what is it? Non-competitive. Non-competitive. Specifically, end product inhibition. And so what could be the biological purpose? Oh my gosh, what could be the biological purpose of this negative feedback inhibition? To not overproduce? Unneeded isoleucine. Conserve energy. And to not waste threonine when isoleucine is available and threonine is needed elsewhere. Threonine has a bunch of functions that it does on its own, and so if we're like wasting it just to make isoleucine when we already have isoleucine, it then can't go off and perform its functions like becoming tooth enamel or metabolizing fat and preventing buildup in the liver. And so it's important that this is regulated, and the most efficient way to regulate it is by end product inhibition, because then it kind of shuts itself off, versus there having to be some other thing that spends energy to come in and turn it off. So it'll get inhibited when there's too much isoleucine. And then this process will start where your threonine is being converted when there's too little. Why did I write that in yellow? Actually, you can see it on camera. You cannot see it in person. So when there's too little isoleucine. And that's it. That's the last page. So I hope you all have a good night. And, yeah. Oh my gosh, my email, stop.